Good afternoon, everyone. I hope you had a nice lunch. I'd like to begin by acknowledging the Yagara and Turrbal people, the traditional owners of the land on which I live and work in Meanjin, as well as the Gadigal people, the traditional owners of the land on which we're meeting today. And I'd also like to pay my respects to all First Nations elders and descendants, and also acknowledge that Indigenous sciences and knowledge has preserved and enabled this land to flourish for more than 65,000 years. So for those of you who I haven't met yet, I'm Beck, I'm a PhD student from UQ, and today I'm going to share some new results, which is all about curing myrtle rust using double-stranded RNA. Cool, so I definitely don't need to spend much time on the background with this audience, but I've just included a few pictures up here of some typical myrtle rust symptoms in and around southeast Queensland, and also the impacts that these symptoms can have on mature trees. So there are three main spore stages of Osteopuccinia: uridinospores, teleospores, and basidiospores, and it can reproduce sexually and asexually. It completes its life cycle on a single host, which really sets it apart from a lot of other rust pathogens, and also has important implications for management. So existing controls of rust fungi include the use of agrochemicals such as fungicides, and also the deployment of resistant genotypes. But these approaches alone are not always sufficient for myrtle rust management, or in a lot of cases also not suitable. So we really need to introduce some new management strategies for this pathogen. So this is where RNA interference, or RNAi, could come in. So RNAi is a post-transcriptional gene silencing mechanism. It's largely conserved throughout eukaryotes. It's triggered by an endonuclease known as DISAR, which recognises these long double-stranded RNAs and cleaves them into short interfering RNAs, or siRNAs. Those siRNAs then form a complex with an Argonaute protein known as the RNA-induced silencing complex. And then this complex then guides sequence-specific degradation of homologous mRNA. So that complex is going to look for mRNA that has the same sequence as that original double-stranded RNA that triggered the process. So we can also co-opt RNAi to defend plants against a variety of plant pathogens, including fungi, and this is generally done through a non-GM approach known as exogenous RNAi. This is where pathogen-specific double-stranded RNA is applied directly to the plant, usually via spraying, and the dsRNA is then taken up into the plant or fungal cell, in this case the fungus, and this process is triggered. dsRNA can also be complexed with a variety of carriers for nursery ex situ conservation or field applications in pathogen management. And complexing with those carriers generally improves the stability so that you have a longer stable window on the plant. So really, what we want to do is use exogenous dsRNA in the Osteopuccinia myrtaceae pathosystem as a management strategy for myrtle rust. And our kind of original and standing hypothesis is that Osteopuccinia processes dsRNA, resulting in silencing of the targeted genes and ultimately a reduction in disease symptoms. So late last year, we published our first paper on this topic, which was mainly acting as a proof of concept that rust fungi, and in particular Osteopuccinia, actually have amenability to RNAi, or to double-stranded RNA rather. And this was a really fantastic collaboration between UQ, the Department of Agriculture and Fisheries, the University of Canterbury, and New Zealand Plant and Food Research. And basically we showed that when we co-inoculated Syzygium jambos plants with Osteopuccinia uridinospores and target gene-specific double-stranded RNA, we saw a significant reduction in disease coverage on the plants. So our next question, to take this kind of one step further into a more applied setting, is can dsRNA be sprayed onto a plant before, during, or even after an established infection to prevent and/or cure myrtle rust? So in our current study, we set out to answer this question by applying dsRNA at 48 hours prior to infection, as well as 24 hours, 6 days, and up to 2 weeks post-infection. So we started with our preventative measures. So for this one, we applied the double-stranded RNA at 48 hours pre-infection, and then we did disease assessments at 2 weeks post-infection. So you can see here from these images that in our treatments, which are the two ones in the green on the bottom, we saw a significant reduction in disease coverage on the leaves. I should also add that the controls are the negative dsRNA and the GFP. GFP is a non-specific control, so they're still receiving double-stranded RNA, but it's not targeting a gene in osteobuccinea. So we would expect to see no impact of the double-stranded RNA. In addition, at this time point, we also sampled some of the plants at 2 days post-infection for scanning electron microscopy, or SEM. So you can see here in this panel on the right that in the controls we saw the Uridinea spores germinating with these long germ tubes and terminating in aprosoria and infection pegs, which were then penetrating into the leaf. And in the treatments, in the lower image, most of the Uridinea spores actually hadn't germinated at all, but when we did see ones that had germinated, they had these really short, stumpy germ tubes with no aprosoria. And also that result validated what we had previously seen in vitro. Cool. So then we moved on to our curative work. So we firstly applied the double-stranded RNA at 24 hours post-infection. At this time point, the pustules have germinated and penetrated into the leaf, but there's no visual symptoms on the plant yet. And after 2 weeks, we saw a significant reduction in disease coverage on treated plants, as you can see here again by the lack of pustules on those leaves or the reduction in pustules. And this disease assessment is just done by measuring the % coverage of the disease on the leaf. This is what I get for using big pictures. Okay, cool. So we also assessed the 24-hour treated plants, how they recovered at 6 weeks post-infection. For this, we used a plant growth scale from 0 to 5. This is just one that we came up with. So it's more of a qualitative scale, but 0 being totally dead and 5 indicating a significant amount of new growth. And with this assessment, we saw a significant increase in plant health. In our treatment plants, we saw much more new growth in the treatment groups, whereas we generally didn't see much or any in the controls, and that's also reflected in the graph on the right-hand side there. So then we extended our curative window out to 6 days post-infection. We chose this time point because this is where you see the first visible symptoms of osteobuccinea infection on the plant. Again, this is just on Syzygium jambos. It's different in different hosts, but in jambos, you start to see this red flecking at about 6 days post-infection. And when we assessed our disease assessment at 2 weeks post-infection, we saw, again, a significant reduction in disease coverage in plants that were treated at 6 days post-infection. And similarly, when we looked at how the plants had recovered after 6 weeks, we saw there was a significant improvement in plant health. And what's really interesting is if you look, do I have a pointer? Yeah. If you look at this one here, which is a beta tubulin treatment at 6 weeks post-infection, you can see that it experienced a bunch of defoliation, so it was still a pretty severe infection, but it's actually recovered and got a good amount of new growth both at the top and down at the bottom there. So that's something that's really cool to see. Even when the infection is quite severe, we're still seeing the plants recover really well in the treatment groups. So we also did a bit of imaging on these same plants. So we sampled for imaging at 2 days post-dsRNA treatment, which was 8 days post-infection. And we used 2-photon confocal microscopy and scanning electron microscopy. So these ones here are all of the controls. In the black and white images, this is our 2-photon microscopy. And you can see here intercellular hyphae and hostoria within the leaf. And then on the leaf surface, we were seeing the production of new sori, new pustules developing with hundreds of new Uridinea spores inside. In our treatments, we still saw intercellular hyphae and haustoria, but maybe a little bit of a reduction in the spread of the intercellular hyphae. But what was kind of really, really cool is that when we looked at the leaf surface, there was almost no new pustule development at that point. And when we did see that a new sorus had started to form, it was kind of, it was kind of inhibited and, okay, I can't go back. It was kind of inhibited and malformed. So it wasn't producing those loads of new Uridinea spores like we were seeing in the controls. So then finally, we extended our curative window out to 14 days post-infection. So at this point, you can see from the image on your left that the plants are experiencing full-blown infection. They're very severely infected. And that's the point where we're spraying the double-stranded RNA. And then we look out at 6 weeks post-infection. And again, we're seeing a significant improvement in plant health and growth on our treatment plants, even in cases where the plants had experienced severe defoliation. So we wanted to look a bit deeper and further kind of quantify this plant health at 6 weeks post-infection. So for this, we looked at some measures of photosynthetic capacity, and kind of from the literature, the two best proxies for net photosynthetic rate are stomatal conductance and maximum yield of photosystem 2. So we went with those. And what we saw was a trend towards an increase in our treatment groups of stomatal conductance. It was only significant at one of the time points at 6 days curative. But yeah, still seeing that general trend. And then when we look at the maximum yield of photosystem 2, again, we're seeing an upward trend in our treatment groups, and also much less variation in the treatment groups, but that was only significant at 14 days curative. And this lack of significance is probably likely due to the fact that for this kind of data, generally you use a much bigger data set than what we would typically use for our whole plant experiments. So here are all of our negative and non-specific controls. You can see there is a lot of dieback, a lot of defoliation. Some of the plants are totally dead. They're not looking very happy. And then if we compare them to our treatments, there's abundant new growth. They've recovered a lot better by that 6-week mark, and they're generally looking a lot happier. So if we go back to that original question, can we use dsRNA to cure myrtaceae of severe myrtle rust infection, then at least in some settings, the answer is heading towards a yes. So just a bit of a summary and a wrap-up. We saw significant reductions in disease coverage in plants that were treated with dsRNA at 48 hours pre-infection and right up to 14 days post-infection. For curative treatments, we saw improvements in plant health and also photosynthetic capacity at 6 weeks post-infection. With 2-photon and scanning electron microscopy, the data we collected suggests inhibitory activity of dsRNA on intercellular hyphae or haustoria, or even at the leaf surface. And these findings could have immediate potential in the management of myrtle rust in Australia in some settings, such as ex situ conservation in industry, for example, in lemon myrtle plantations, or in the nursery industry, or in the treatment of individual trees or small populations. And that's pretty much it for me, but just one note to leave you with that I was just thinking about before. I think it's really important to remember that even with all of these exciting new management opportunities, there's not going to be one single treatment that's a silver bullet, and really we're going to need to embrace a kind of multi-pronged collaborative approach if we want to stand a chance. So I think it's important to think about when we hear about all these new management strategies over the next few days, how they can work together and kind of what settings we can we can use each of those strategies in. Yeah, that's all from me. Thank you so much, and happy to take questions.
Okay, do we have any questions for Beck? We've got a couple. Yep, we've got one in the back there, if we can get a mic. Yeah, sorry. Can we just get a microphone back at the back there?
Longer than that time frame? And if so, did the effects of the double-strand RNA wear off? I'm interested in how long it's going to last. Kia ora.
So I still have the plants, and generally I would keep them for like a couple months after the experiment, just kind of in case, but I haven't quantified anything past 6 weeks. In general, you continue to see that the ones that were doing well continue to do well. With this particular species, Syzygium jambos, they're very, very hardy. So after a certain amount of time, particularly if you're cutting back and fertilizing and stuff, you would, I think, start to see the controls probably catch up, because they just can bounce back from pretty much anything most of the time. But in general, just in the weeks that follow, we continue to see that the treatments are doing much better, but again, haven't quantified it, so yeah.
Thanks. Robert Beresford from Plant and Food Research in Auckland. I'm just wondering if anybody shares your optimism sufficiently to pick this up and run with it as a commercial proposition, or to develop a sprayable product?
I would say yes, there are people that share the optimism. Again, like I just said, I think it's really important to remember that it's not appropriate for every setting. But, I think, for example, people in the nursery industry, just at nurseries that I've gone to, they have like a huge amount of excitement for this kind of technology. So it's kind of is in the works to get it to a more commercial or applied stage, but yeah, it's still got a while to go. Yeah.
Okay, thank you.